So welcome everybody. So I'm here to introduce Dr. Fred Sheldon, and it's an honor for me because it's usually the other way around where Fred would be introducing me, but now I get to introduce Fred. And what better way to do it than with a story, because that's Fred loves stories, and I'm gonna start off with a story. And so my second semester here, I was able to go on this long road trip with Fred up to the Northeast, visiting all these museums. And there I learned a lot about Fred's history because we visited all of his former haunts. So we stopped in Maplewood, New Jersey, where there is a train station. With Fred told me that he used to work as a painter and he painted this train station. And we were marveling this train station while waiting for our train to take us to the American Museum. Next, we stopped at the Academy of Natural Sciences in Philadelphia, where Fred had worked as a curator before he started here in LSU. And we were able to dig through this wall that had been hidden up, where all the former curators had a picture of them. And so we were trying to search where Fred's picture is, and it's in the corner somewhere, just hidden along there. We also stopped at Yale University, which is Fred's alma mater, where he did his bachelor's, master's, and his PhD work, and a lot of which we'll be hearing about it when Fred gives his talk. And so I learned a lot about Fred, and I'm so glad to have been his a final PhD student here. I learned a lot. His name is synonymous with birds of Borneo. And so without further ado, I'm just gonna ask Fred to give his talk, and we'll learn a lot more about his life and his career. I'm gonna end this by saying that Fred has been a curator of birds for longer than I've been on this earth. So that should tell you how long this legacy is.
Thank you, Subir. That was eloquent, wonderful, and not too embarrassing. I usually think several times a year that when students look at me, they say, how did this old fart get here? Why is that guy a professor? I mean, if he can be a professor, anybody can be a professor. He doesn't know R or how to use to program anything, or to use a pipette, or he certainly couldn't do a genomic pipeline. And worst of all, he has a Kardashian index of 0. So he's obviously a 0. So what I thought I would do is tell you how I got here. This is very selfish of me, but I'm going to provide a history of molecular systematics in the 20th century, stopping at the 21st century. So stopping when you all came on board pretty much. And I'm going to start a bit wide, but I'm going to narrow the topic down until I appeared on the scene. And then I'm going to talk about my small contribution to the field and some of the harrowing experiences I went through in the process of coming to my present situation. First of all, why molecules? All right, there's 3 reasons why we use molecules. They add extra characters. Morphology can only do so much, so molecules give you more characters. Molecular characters are closer to genes. You know, when you're looking at morphology, you're looking at stuff that has gone through weird developmental processes that make it very difficult to interpret. And in principle, molecular data are objective, quantitative and objective. But I'll show you that that's not always the case. So let's first look at the old-fashioned way. Here's a distinguished LSU alumnus, Joel Craycraft, now working at the American Museum. But back in 1974, he worked at the University of Illinois, Chicago as an anatomist, and he did a study of the old-fashioned way, looking at bones of the paleognaths. Now, the paleognaths include the big running birds, the ratites, ostrich, emu, cassowary, rhea, like that, and some other stuff. Okay, and this is the tree that Joel came up with back in 1974, looking at bones. And this is the tree that the early bird group came up with, looking at molecular characters more recently. And it's a better tree. So I'm just going to tell you briefly why Joel did such a bad job on this tree. And it's really not entirely his fault, but part of it is. So let's take a look at this. There's a tinamou. Now, a tinamou is a paleognath. It has a paleognathous palate. So it's, it's very closely related to ostriches and rheas and things like that. But it flies. So if you're looking at morphological characters and trying to figure out how big non-flying birds are related to a flying bird, you're going to get it wrong, and he got it wrong. You can see he has the tinamou out there where you'd expect flying birds to be away from those big guys. There's a kiwi. Kiwis live in New Zealand. There are no native mammals in New Zealand.
Yes, there are.
There are no native mammals in New Zealand. What?
Bats.
Oh, that's not a mammal. Leather. But a kiwi is a bird that's become a mammal. I mean, it's furry, it digs for worms, it runs around, it can't fly. It's basically a mammal. So of course he's going to get it in the wrong place if he's looking at morphological characters. And he did. A kiwi is closely related to emus and cassowaries. So, you can't really blame the mistakes too much on the morphology because it's tough using morphology. Now, there was another element to this study, though, which is that Joel was trying to support his buddies at the AMNH, like Don Rosen and Gareth Nelson, who were Vicariant biogeographers. I can see Prasanna's going, oh. And of course, he's going to come up with a phylogeny that's exactly the same as the Gondwana breakup that explains the distribution of these birds by vicariants. So that's another reason that's wrong. That's the issue of objectivity. Molecular systematics goes way back. I think this is the coolest stuff. This guy, George Henry Faulkner Nuttall, did many incredible things. He worked on malaria and he did all kinds of stuff. But he wrote a book in 1904, which is in the LSU library, on basically immunological studies of vertebrates. And basically what he did was he took a protein from an animal, pumped it into a rabbit, bled the poor rabbit, got the antibodies produced by the rabbit, and then reacted those with the original animal and anything else you wanted to compare. And he had a pretty cool method. Basically, when he did this antigen antibody reaction, he got precipitate. And so here was his control, which had the greatest reaction, of course. And then there's a substantial reaction between X and C and a pretty good reaction between X and B and so forth like that. So he would normalize those measurements and come up with a table. And then he would repeat the whole process by pumping a different species into the rabbit and collecting the antibodies and so on and so forth until he got a similarity matrix looked like that, basically showing the similarity or the relationships to his mind of the creatures he's looking at. It was pretty good. 120 years ago, the guy did this. And let's take a look at some of his data. He pumped some chicken into that poor rabbit. And I say poor rabbit because I used to have to bleed these rabbits to gather the antibodies. It's a horrible thing. Anyway, so here, this is just to show you that back 120 years ago, he figured out that by these reactions, that the ducks and geese are the closest relatives of the galliforms, the chickens. So that was pretty amazing, really, since we now know that that's true. And in the title, you may have noticed that I'm going to talk about hominoids. And so here's my introduction to hominoids. He also took human proteins, pumped them into the rabbit, collected the antibodies, and reacted them with various things. And he reacted them with the great apes, chimpanzee, gorilla, orang, and he got exactly the right relationships. Man's closest relative is chimpanzee, then comes gorilla, then comes orang, then comes various monkeys. So he had it right years ago. And of course, I'm going to come back to that story because I was involved in some more recent studies of these great apes. By the 1960s, I skipped over 50 or 60 years of stuff. By the 1960s, people were doing a lot of these molecular comparisons, and they were having annual meetings to discuss their various approaches. They did immunology, of course. They were still doing immunological comparisons, protein electrophoresis, protein sequencing, but they also looked at all kinds of other stuff. And I made a little list here. We got bile salts, plant secondary chemical compounds, polytein chromatins, lipid metabolism, venom constituents, lens proteins, et cetera, et cetera, et cetera. So in these meetings and in these books that were produced by the folks doing molecular taxonomy, proteins were the big thing. And proteins were very important because of these two characters, Emil Zuckerkandl and Linus Pauling. They did early work comparing proteins, and they really pushed the idea that proteins are close to the gene, as I said at the beginning, and they came up with this term semantides. So a semantide is a molecule that's close to the gene. So you've got your DNA and then RNA, that would be a really good semantide. Proteins 2 steps away, that's a good semantide like that. As opposed to other molecules that go through more developmental or other such processes that make them a little less, less sort of reflective of a gene. The other thing they came up with was the molecular clock. They did some comparisons and they said, wow, it looks like these proteins change in a regular pattern. Here's some of the data for that thinking. This was produced by Emmanuel Margoliash at Northwestern. So Emmanuel spent about 10 years sequencing proteins, and he finally got a man, horse, pig, rabbit, chicken, tuna, and baker's yeast, an odd list of creatures. But when he compared them, he would get a genetic distance. Now I'm going to emphasize genetic distances because that's what I worked with when I was a graduate student. And that's what a lot of the stories in this talk are going to come down to, understanding genetic distances. So I'll kind of beat that into you for a while. So the genetic distance between a horse and a man, that's the number of amino acid differences, is 12. And the genetic distance between a horse and a pig is 3 amino acids. So I would say from that, a horse is closer to a pig than it is to man. Is that right?
That is true.
Okay. All right. The other thing that he did was calibrate these things. Here's where the molecular clock comes in. They had fossil data for the divergence between a horse and a man, or at least between a horse and, they had some date of the earliest horse or the earliest man. Anyway, and so from those dates, they calibrated these amino acid changes to 1 amino acid change per 10 million years. So they actually had a clock. That was a clock. Now that was good. That's quantitative. It's objective, amino acid differences, but it was slow. So that frustrated folks. So they tried to find faster techniques. And now I will introduce my professor, Charles Sibley, who in those days worked at a second-rate university. But he was doing state-of-the-art work by electrophoresing various proteins. So here's an example of a diagram from one of his papers in the 1960s. Showing the electrophoresis of egg white protein on starch gel. And so they would load the protein there and then run electricity through it, and the protein would run according to its molecular electrochemical properties. And that looks all pretty good. And so he would make comparisons. Well, the comparisons don't look so good. They look pretty ugly. In fact, they're almost completely uninterpretable. So although this was a fast technique, it was a very rough and really not very quantitative way of comparing proteins. Immunology was still going strong. So in the mid-1960s, microcomplement fixation was introduced. This was a vast improvement on the old immunological techniques that allowed you to measure just the difference between proteins that differed by 1 or 2 amino acids. So it's the same thing. The poor rabbit is still involved, except that you're reading the reactions in spectrophotometric tubes on a spectrophotometer. And so you're getting pretty good numbers and pretty good similarity values. And that's what people did. And then from the similarity matrix, they would make a matrix of immunological distances. So the distance from X to itself is 0, no distance, X is X. The distance from X to A would be 100 minus 45, which is 55. X to C would be 30, and so on and so forth. So they got a distance matrix like that. And from the distance matrix, you'd get a tree. Now, the people who popularized this microcomplement fixation methodology were well known to Mark Hafner, his pals at UC Berkeley, Alan Wilson, a biochemist, and Vincent Sarich, an anthropologist. All right, so those guys compared the hominoids. Again, I don't really care about hominoids, but because it's important to getting to where I was in the history, I got to tell you about hominoids. And so they got immunological distances of these hominoids, and they drew a tree. And the tree was pretty much like the one that Nuttall got, 60 years earlier. They didn't actually resolve the human-chimp-gorilla trichotomy. But what they did was they calibrated the immunological distance. So we just saw that Margoliouth calibrated the amino acid differences. These guys were calibrating the immunological distance, and the fossils indicated a divergence between monkeys and the great apes at 30 million years. And by carrying that forward, they got the divergence of the human, chimp, and gorilla at 5 million years. Now, to us, that's no big deal. I mean, so what? But apparently that's a big deal for anthropologists. From the bones that they were digging up, they had the divergence date between humans and the other great apes much older, 10, 15 million years. So this was a big deal, and it really put these two guys on the map. In fact, I would say that Alan Wilson went on to be the greatest molecular systematist of the 20th century. Okay, the other thing they introduced, that was a science paper. Here they've got a PNAS paper. They introduced the relative rate test. So the relative rate test is a distance comparison between an outgroup A and a couple of in-groups. So the distance, if the distance from A to B is the same as the distance from A to C, you guys can see my cursor, yes, then there's a molecular clock. And that's what they did. They did a whole bunch of these things, but here's one where they compared monkey to the apes, and they got a distance from monkey to each of these apes of about 2. There's 2, 1.95, 2.13, like that, within their error. So that supported the molecular clock. And those two guys were adamant supporters of the molecular clock for the rest of their careers. Wilson and Sarich. Now, when I skinned my first birds, what I was given were frozen birds from the Alpha Helix expedition. So that was 1976, and I'm skinning these horrible shriveled birds that Sibley had collected in this expedition in 1969. They'd been sitting in a freezer. Sibley had moved up the academic totem pole, to a worthwhile university. He actually was the first William Robertson co-professor at Yale, which is the position that Rick Prum now has at Yale, for those of you who know Rick Prum. So anyway, Sibley moved to Yale, and among the early things that he did there was raise money from NSF to hire this boat from Scripps to go to New Guinea and serve as a base for a collection of animals and a lab for comparisons of proteins of those animals. And on that expedition were Alan Wilson and Vincent Sarich, who we've just met. And so they were on that trip. Also on that trip here, Herb Dessauer, a great hero of LSU, professor at the medical center in New Orleans for 30 years or 40 years, something like that. He was a herpetologist, and he had a huge herpetology collection, a collection of tissues for herpetological research, which he and Mark Hafner got together, put in our basement here in this, of this building, as the founding part of our now world famous tissue collection. So that's Herb, that's the guy who gave us all those herpetological tissues, which people still are after all the time. And some of them came from this expedition to New Guinea. I would also say that Sibley being Sibley, got in a fight with the captain of the ship, so they weren't speaking, and became mortal enemies with Alan Wilson and Vincent Sarich, which becomes important later on. All right, so, all right, proteins are fine. These guys are all doing proteins. They didn't have DNA sequences. They didn't have DNA really until some folks started hybridizing DNAs. And the first people to do that were McCarthy and Bolton in 1963, and they compared various bacteria. And so they would take one strand of E. coli DNA and say, compare it with a strand of, by hybridizing it to a strand of some other kind of bacteria. And they would get these basically degrees of hybridization is what they got here. So E. coli hybridized with E. coli was normalized to 100%. And then they got these various things. E. coli with Proteus vulgaris was 14%, if I'm reading that right. And E. coli hybridized with a mouse DNA was 1%, not very much. And so E. coli is not particularly closely related to a mouse. Okay, so Sibley tried this. Charles Sibley, the guy we just met, tried to do this in 1964. It didn't work. And the reason it didn't work was that nobody knew that vertebrates, birds, which is what he was interested in, had repetitive DNA. And when if repetitive DNA is in there, you don't get meaningful distances. And if somebody wants to know about that, I could talk about it at another time. But this guy, Roy Britton, figured out that there was repetitive DNA, and he also figured out how to get rid of it. And so once that happened, it was possible to do DNA hybridization. So because that's what I used for my PhD thesis, I am going to bore you now with a short, but I hope clear explanation of the process. All right, so you take single copy DNA, you get rid of the repetitive DNA, you radiolabel it, usually with something really horrible. We used I-125. You guys are so lucky nowadays. They are so, it's amazing that I'm 70 years old. I mean, it's just incredible. All right. Okay, so you take DNA of, where's my mouse? All right, you take the DNA of one creature, you radiolabel it, you hybridize it with itself as a control. You do that at about 60 degrees. This is, those of you who've done PCR, this is where it all comes from. You thought that it was invented more recently, but it was invented in the 60s. This is where it all comes from. And then you dissociate the hybrid, slowly taking samples, and you figure out its sort of average melting temperature. And you normalize that. That's a genetic distance of 0. So you've hybridized, this is your homoduplex, this is the same species hybridized. So That's 0. Then you take your radiolabeled DNA and you hybridize it to a different species. And of course, it's going to have lower complementarity in the DNA because they're different species. It may melt at a lower temperature, let's say 5 degrees lower than the homoduplex. So you get a genetic distance of 5. And then you repeat it with species 3 and with species 4, and you get genetic distances right there. So there they are. This is what's called an obligate genetic distance process, because all you get is distances. All right, then you, like as with immunology, then you got to go back through, you got to radiolabel, you get single copy DNA of species 2, radiolabel it and compare it all the way around. And you'll get a complete matrix of genetic distances. And from that matrix of genetic distances, you build your tree. Very simple. Okay, just in case you're interested, so you make your hybrids here at 60 degrees, and then you start heating up the hybrids, and you take samples at various temperatures, and your homoduplex is going to melt on average at a higher temperature. This is the median temperature versus the other things that you compare. And then you get, this is the genetic distance along that line. Or you can take the point at which most of the hybrids melt, which is the mode of that melting curve, whatever. So basically that's where the data come from. All right, so after that long preamble, and Subir took a lot of time, so I'm not counting that against myself. I'm finally coming to me. So here's my professor, Charles Sibley, and there is his research associate, John Alquist, who recently died. I wrote a memoriam for the AUK about John, which I suggest, if you're at all interested in this talk, I suggest you read it because it tells you a lot about what these guys are doing, but I'm going to tell you here as well. So in 1975, they started hybridizing DNAs. In 1978, I arrived at Yale as a graduate student. 1980, they published their first paper. So it took them 5 years to publish their first paper. So that's a lot of work. Then I quit, and I'll come back to that. 1980 to 87, they published 33 bird papers. In 1984, they published a hominoid paper. That was a big mistake, but that's where the hominoids come in. 1984, I returned. 1986, Sibley was elected to the National Academy of Sciences, and he retired. And I finished my PhD. 1987, they published their second hominoid paper. That was a really big mistake, as we'll see. And then in 1990, Sibley published his magnum opuses, which were a classification of birds with John Alquist, and I'm sorry, a description of all the DNA hybridization stuff with John Alquist, and a classification of birds with Burt Monroe, LSU PhD, 1965. And Burt actually wrote that entire classification book, but Sibley being Sibley, he's the first author. All right, so why did I quit? Well, Charles Sibley was a very difficult person, which is why he got at loggerheads with the captain of the Alpha Helix and Alan Wilson and those guys. And here's a sort of typical example of a conversation I might have with Sibley. This one was about some collecting expedition work in Australia. And I would say, well, Dr. Sibley, I don't really think that's right. Don't you talk back to me, you son of a bitch! I'll ruin you. You'll never work in ornithology. Needless to say that a few of those conversations and I was irritated. So I quit. And poor Jody, you know, I'd come home and go, I quit graduate school today. And, this after support, she gave up her career. She moves to a different state, and then I give up the whole thing. It was bad. All right, so let me describe Sibley and Ahlquist. In order to understand my contribution, you got to understand Sibley and Ahlquist's modus operandi. And the first thing was they adamantly believed in the molecular clock. Sibley, we would have lunch every day. Sibley worked at home in the morning, and he'd come in for lunch, and then everybody had to sit and have lunch with him. And we had a big table in the bird range, and he would pontificate. And for 3 or 4 years, it was always the same thing. The average rate of nucleotide substitution measured across the genome and over time is the same in all lineages of birds. It's the same. Okay, we heard that a lot. And they published lots of times these kinds of diagrams. Here's a genetic distance, a DNA hybridization distance versus time of done by some other folks of sea urchins and the divergence of orangs from the rest of the great apes, and that line would show that there's just a constant rate through time. Of course, this was all based on fossil dates, but that was one line of proof, and the other was an endless series of these relative rate tests where he would show that the outgroup is the same genetic distance to the in-group, and all the outgroups are the same genetic distance to the in-group. That's why I went to the trouble of explaining to you before, because you have to understand that. Okay, so that's one thing. Everything was that there was a constant rate of evolution, and the reason that Sibley that he did that is he pushed it so hard as he thought it was required for DNA hybridization. Okay, so the other thing that those guys did was they published in bird journals and books. They never published in Evolution or Cisbio, or in those days it was Ciszo, or any of those things. They stuck to these kinds of lower level publications. They never provided raw data. They just produced distances, showed you the distances. And they drew their trees by hand. So they weren't using, Bayesian, wandering models or any of that stuff. So here's some typical data, a typical study. So this is actually one of the best studies that Sibley and Ahlquist ever did. And it was because it was right in the range of DNA hybridization's resolving power, showing that starlings and mockingbirds were sister groups. So it's pretty cool because starlings are basic old world birds, mockingbirds, new bird world. The whole thing was pretty cool. And so here they go. Here are the genetic distances that they would publish. They would just make a list of genetic distances like that. And then they'd come up with a tree. So I'm just going to point out that, and John Ahlquist did this. John Ahlquist computed all of the genetic distances by hand on a calculator. And then he would draw the trees by hand, and he would just literally interpret those genetic distances. So here's this glossy starling here, and the distance to the next bird was 1, and he would put 1, and then the distance to the next bird it was 2, and he did 2, and then it was 2.5, and like that. And that's why you see the trees are the comb shape, because he just literally interpreted those distances. And then they would label, you can see the things that are labeled by the dots up there. Maybe you can see it. They would label a couple of the things, and that would give them a little bit of structure in the tree sometimes. But that's how they did it. They just by hand, the old-fashioned way. Now, most people who had no stake in this thought it was fantastic. Stephen, let me think about this. Gould. What's Gould's first name? Stephen Jay Gould. Stephen Jay Gould would write these papers in the, these glowing letters in the American Museum Public magazine. How great this was. So a lot of people thought this was fantastic, except a lot of people who actually cared about bird systematics were not impressed. And here's one guy who was not impressed. This is Storrs Olson. Now, Storrs Olson wouldn't know a molecule from an elephant. He wouldn't, he didn't know any statistics. He was the quintessential old-fashioned seat of the pants Ernst Mayerian taxonomist. You know the relationships of animals by knowing the animals. So that was his background. But he was smart enough to see that Sibley was not doing it the right way. So just to read a couple of bits, your latest paper in EMU, that's the Australian Bird Journal, strikes me as nothing more than unalloyed propaganda. In my opinion, sir, you are quite simply a fraud, a snake oil salesman who resorts to precisely the same tactics as the creationists. Whereas you can hoodwink a legion of bird watchers and hacks like Stephen Jay Gould, you have not earned the respect of those blah, blah, blah. Okay, just a classic. Van Remsen put this letter in my mailbox. All right. So in 1984, Sibley just decided he would tackle the hominoids, and to his credit and Alka's credit, this was a good, thorough study. They radio labeled all the taxa. They went every which way. They had multiple comparisons of each different individuals, and they came up with this tree that clearly said that humans and chimps were sister groups, and that the gorilla was the next closest thing. And lots of people thought that was cool, but Sibley had never tackled with the anthropologists. Anthropologists are really sensitive people, and they were not happy, and a lot of other people weren't happy. Okay, and they just attacked the heck out of that, and I'll come back to that later. They were not happy. Oh, he also made the mistake of publishing in the Journal of Molecular Evolution, a high-quality journal. I don't even know if the Journal of Molecular Evolution still exists. It was like the go-to journal in the 1980s. But anyway, they made the mistake of putting it in a place where lots of smart people saw it. All right, what else was going on in systematics? Oh, lots of great stuff. So the cladists were fighting the statistical tree builders. So classic Steve Ferris versus Joe Felsenstein. The morphologists, the morphocladists, were fighting the molecular people. They didn't like the molecular people. Okay. So to understand why the cladists disliked the molecular stuff, and particularly DNA hybridization, I have to give you a little background in cladistics, and I hopefully some of the folks who are visiting don't know this, and it's worth the process, but I'll quickly get through it. So recently there was a group of birds called the Mirandornithes, which was discovered, which consists of grebes and flamingos, very unlikely group. Okay. And the way a cladist would study to discover those relationships, they would do what's called a character matrix. So they would have the taxa on one side, flamingo, grebe, painted snipe, rhea, and ostrich. Those are the taxa we're looking at. And they would have the characters over here, of various types, okay? And then they would take those characters and they would put them on a tree according to the matrix. All right, now, two of those characters, 11 primaries and chalky eggshells, apparently are particular to grebes and flamingos. So that's what's called a synapomorphy. Characters that support that group, okay? Keeled sternum. Well, keeled sternum is a synapomorphy. It's a shared derived character of all flying birds if you're comparing them to rheas and ostriches, which are not flying birds. So those are synapomorphies. So they use those synapomorphies to identify monophyletic groups. But a keeled sternum is a plesiomorphy when it comes to trying to figure out the relationships among the painted snipe, the flamingo, and grebe. All those birds have that character. It doesn't tell you the relationships in that group. So it's kind of, it's homology, but it's useless in terms of phylogenetics. Feathers is another one. Feathers defines birds more or less, but it doesn't, it's a plesiomorphy for birds. It doesn't tell you how individual groups of birds are related to each other. And then there's one other issue, which is homoplasy, which is the noise in the tree. So it happens that painted snipe and rhea in those groups, the males take care of the chicks, or they take care of the nested chicks. Okay, so that's a convergent character that's noise. It doesn't show you anything about relationships. It's ecological. All right, and the way that cladists figure this out is they get their character matrix, they try the characters on all different topologies, and they come up with the tree that's the shortest tree. And that's the Occam's razor, the principle of parsimony. The simplest explanation is the best. All right, so cladists versus people like Sibley who use genetic distances. The cladist complaint is that genetic distances measure overall similarity. They don't differentiate between apomorphy, which is useful, and plesiomorphy, which is not useful. Okay, so that, and I heard that complaint many times. And here it is, it's sort of like that. Here's the microcomplement fixation thing. You get a similarity matrix for sure. And that definitely mixes plesiomorphy and apomorphy. But that's not how you build the tree. You build the tree by getting a distance matrix, which is about distances, not similarity. And from those distances, you get a tree. And so it's based on apomorphy, not plesiomorphy. Just in case you don't know that, Prasanna. Poor Prasanna has to put up with me because I was born in this contentious era, or I was raised in this contentious era of cladists versus molecular systematists. And he comes ultimately from the American Museum, which is a center of cladistics. And so even though he's a different generation, I constantly beat on him. I'm sorry, I take my apology. All right, what else is going on in systematics? People are using allozymes, lots of allozymes, but most of them are PopGen, population genetics. Although Scott Lanyon, who is here and is now a distinguished professor at the University of Minnesota, he used isozymes for phylogenetics. People were using mitochondrial RFLPs for PopGen. And one thing about the mitochondria, it was discovered to evolve really fast. It was discovered by this guy, Wes Brown. I scouted around for a picture of Wes. This is the only one I could find. He looks like an eco-tourist who got caught taking a leak. Anyway, Wes worked with Alan Wilson. Here's Alan Wilson, a guy, as I said, he's the greatest molecular systematist of the 20th century. So Wes worked in Alan Wilson's lab and discovered this fast rate. And basically here's some evidence. The distance between humans, say, and monkeys is about 13 different monkeys, 13, whereas the DNA hybridization distances are much smaller. So DNA hybridization, the nuclear DNA from which DNA hybridization is derived is evolving much slower than mitochondrial DNA. This is great study. And it put a big dent in the molecular clock because, hey, wait a second, we're showing right here that different parts of the genome, different genomes are evolving at different rates. But that didn't really affect Sibley or Alan Wilson for that matter. They just said, well, mitochondria are different. They don't have repair mechanisms. It's not the same. In nuclear DNA, molecular clock, period. The other thing that's really interesting here is the compression in these mitochondrial data. You can see differences between monkeys and differences between humans. They're all around 12 or 13. Whereas the nuclear data, the distances get larger as the genealogical relationship gets larger. So they're not compressed by back mutations of nucleotides, which is really important. Okay, I still haven't talked much about myself, but now it's going to be totally self-centered. Where was I when this was happening? I was in Borneo, so I quit. And Jody and I went off to Borneo for 3 years and had the greatest time of our life. But I had done a lot of work on my PhD, so I came back. As my Australian friends, I came back and licked Sibley's boots and finished my PhD. And I did a phylogeny of herons. There's the phylogeny, published in the author. The other thing, though, when I was doing that PhD is that I discovered that different lineages of herons were evolving at different rates. There was no uniform average rate of nuclear DNA evolution. The bitterns were evolving fast, and tiger herons and boat-billed herons were evolving slowly. And when I first went to Sibley with that in about 1984, that's crap. That's just experimental error. You know what we do in that situation is we force it. We force it. Make those branches the same length. Alan Wilson reviewed my ALK paper, which wasn't this paper, which was about the rates, but I did mention the rates, and he just said, it's not true. It's not true. But anyway, this put me on the map. I remember I was ecstatic that David Swafford said, "You're Fred Sheldon." We read your paper in their discussion group, wherever he was, Illinois Natural History Society, whatever. Anyway, so there were highlights to that paper. It was the first direct demonstration of variable rates of genomic evolution, not depending on fossils. Fossils were always the weak link in studying rates. No fossils. It was the first demonstration that closely related organisms evolve at different rates. People knew that pretty well that rodents evolve faster than hominoids, their insects were faster than vertebrates. They sort of knew that from fossil data. They had no idea that closely related creatures could evolve at different rates. It was the first application of a ratio test. This drives me crazy. 10 years after this, nobody ever contacts me. I don't know why this is. Hielsenbeck and Bull came up with the ratio test, and they published a paper in Science, and it was a big thing, and I mean, I did that 10 years before. They didn't even cite my paper. And I clearly explained why genetic distances, particularly unsaturated DNA hybridization distances, work. By the way, just a little inject, much later, while I was here at LSU, we showed that cytochrome B, or mitochondrial DNA, has the same rate characteristics as the nuclear DNA. The bitterns evolve fast, and the boat-billed herons and tiger herons evolve slowly. That was, I didn't expect that. I wasn't even sure it was true when I first did it, but now I'm pretty sure. Okay, so this presented a problem for Sibley, because for years he'd been saying there's the uniform average rate of evolution, and he had drawn thousands of trees that look like that, where all the branches are perfectly even. And all of a sudden, it's like, and he said to me, the scales have fallen. He understood it, but because he had been worried that it wouldn't be possible to draw trees if he didn't have a clock. And you certainly couldn't label as few tacks as he did in drawing trees. So magically, they came up with, well, we've been correcting the data all along for generation time. We know that small birds have faster generation time, and therefore their DNA turns over faster, and so they have a faster rate of evolution than big birds, which have a slower rate of evolution. We knew this all the time. We've been dealing with it. Now, of course, that doesn't explain, why he'd been saying there was a uniform average rate, nor did it explain why these guys had how these guys corrected that. Even in Sibley's book, long after this stuff came out, I mean, they never came up with some metric for correcting the data. And of course, they didn't correct the data. Ahlquist just drew the trees by hand according to the distances. And we'll see more about the distance. Oh yeah, we're coming to that right now. Okay, so after the first hominoid study, Sibley, I really hate people who talk more than 45 minutes. This is going to take a little longer. Hang on. So after Sibley published his first paper, he was really irritated that the anthropologists were everybody's giving him a hard time about his tree. So he said to Ahlquist, we're going to do this again, and we're going to have more comparisons. It's going to be undisputable. And so they did. They repeated the whole process, and they came out with another tree. And at this point, I had finished my PhD, and Sibley, Sibley knew that I was using computers to get the distances, computer programs to get the distances, and to build the trees. And so he asked me to, why don't you just run these data through? And so I did. And when I ran the data through, it was perfectly clear that the DNA hybridization data did not resolve this human chimp gorilla trifurcation. And I came to Sibley's office and I said, Charles, before that I had to call him Dr. Sibley, but now I had a PhD, so I called him Charles. Charles, I'm sorry to tell you, but these data are fudged. I mean, John just changed the data so that they would, break the trichotomy. And he got really mad and he stormed out of the office, called up Alquist, talked to his wife and everything, came back the next day. And he said, no, I talked to John and he says that all the manipulations that he did were perfectly logical and so on and so forth. And he said, you know what your problem is? Your problem is that you're afraid you're going to get caught. And I said, yes. Yes, I do not want to be part of this project. I mean, this is crazy. And after that, I never spoke to him again, basically. And he did get caught. Vincent Sarich got a hold of some Sibley and Alquist data, not the hominoid data, but some data. And the guy was really smart. And he figured out exactly how Alquist, and he looked at the data and he had looked at the papers that the data was associated with. And he figured out what Alquist had done and how he had changed the numbers to produce the distances that he produced. And there was a huge blow up, which is I could talk for an hour just on that. We had the NSF funded a special meeting for us at UCLA where we all went and talked about this. Anyway, Sarich knew that I had the hominoid data and he kept calling me up and said, you better give me that data or I'm going to ruin your career. These guys all want to ruin my career. But these guys were such jerks. I mean, Sibley was a jerk. Sarich was a jerk. That normally I'm a pretty hyper guy, but I was just like, whatever. But I wouldn't give them the data. I said, they're not my data. You want the data? Get them from Sibley. They're not my data to give out. Okay. So after that, we're coming sort of toward the end. So hang on back there. These kind of methods, these distance methods were dying. I mean, and the reason they're dying is relativity. In order to get those distances, you have to compare everything to everything else. And whereas when you're dealing with sequences, you've got a fact. A sequence is a fact. You have it forever. You could take one of those DNA sequences there and use it 100 years from now, and it still works. So that was a big problem. The other thing is that sequences were more flexible. Distances, you can only make distance trees, whereas with sequences, you can make distance trees, you can make parsimony trees, you can make maximum likelihood trees, you can do a lot of things. The other thing was fast sequencing. Fast sequencing was on the move, and the first bird DNA sequence paper, cytochrome B paper, was produced in 1991 by Scott Edwards. That pretty much put the nail in the coffin of the distance methods. Well, that was hard for me to swallow because these guys, now NSF was only funding people who were looking at one gene. I was looking at a freaking genome, one gene, and I knew from John Avis that there was a gene tree, species tree problem. Somebody might have heard of that, but no, so that was the end of that. However, I had a few trees in my pocket, few studies, and they were really good. They were accurate trees, which people hadn't, until that time, people hadn't really produced. And at that point, people were saying the tree building isn't important. I mean, you need the tree, but once you got the tree, then you can put it to work to study the evolution of behavior or the evolution of ecology, And there were tons of people coming here to the LSU asking for our tissues so they could build trees so they could study ecological and behavioral issues. It was a huge explosion in the field of systematics, and which is understandable. That's the whole reason for systematics, is to study ecology, to study evolution, to study behavior, all that other stuff, not to build freaking trees. So anyway, I had trees. So what I did was I joined up with my buddies and put those trees to work. Here's a tree that I had, a DNA hybridization tree of the relationships of swallows, which showed that the mud nesting swallows, like barn swallows, were all related to each other. And that actually there was a nice, interesting evolutionary story just about the development of mud nests, and that the whole adopting swallows, like tree swallows and purple martins, were all part of a group, and that the swallows that dig holes, like bank swallows, are the more ancestral swallows. It was a very nice study. Got a lot of attention. Still gets cited, 5 times a year. Also with Frank Gill, before I came here, I worked at the Academy of Natural Sciences with Frank Gill, famous ornithologist. We had a titmouse. I had a titmouse and chickadee phylogeny, a DNA hybridization phylogeny, which we used to study the relationship of the seed cachers. So when a chickadee comes to your bird feeder, it takes the seed away. Most of the time it hides it. Well, it has to have a big brain in order to remember where it hid it. It has to have flocks. It has to form flocks to protect the caches where it hid the seeds. All that stuff led to great success of the seed caching chickadees, which happen all to be related to each other, which was kind of interesting, versus the more depauperate lineage leading to the non-cachers, the famous blue tits and great tits like that. So that was a pretty cool study. Again, using those old DNA hybridization trees. Here's another one with the heron tree, the first tree I showed you. Okay, we studied heron song. You wouldn't think that herons sing. They don't really sing. They grunt, sort of. But there is phylogenetic information in their song. And also we noticed that there was, that the songs, the frequency of the song varied with habitat. If you live in a dense habitat, you gotta sing lower notes, stuff like that. So we did a lot of that kind of thing. And there was, as I mentioned before, there's Sanger sequencing. We did the first Bayesian study of birds. It may have been the first phylogenetic study with Bayesian methods, I'm not sure. But it's definitely the first Bayesian study of bird relationships right there with Linda Whittingham, my postdoc. And here's a nice graph from that study, which shows that the branch support from bootstrapping and the Bayesian support are correlated, not surprisingly. This was actually Adam Lachey's idea, this tree. I felt bad after the fact that I didn't include Adam in this paper, but I made a few mistakes too. Anyway, that was that. And we made the first well-sampled bird family tree. People used in those days, they'd compare 10 fernariids, and they'd say the phylogeny of the fernariidae, Now, it was common, mostly Europeans. But okay, there it is. This is a swallow tree. I mean, David Winkler and I went around the world and we collected almost all the species of swallows and put it into that tree. So it was a nice, complete tree. All right, so now I'm at the end. We're in the 21st century. I decided when I was 50 years old, so 20 years ago, that I became an ornithologist so I could go in the forest, not so I could go in the lab. And so I made a concerted decision that that's what we were going to do. And I went back to Borneo. So there we are, back in Borneo, and I've been there for 20 years. And Subir talked about that. Subir has been one of my great helpers in that process. And that's it. I'd like to thank. What?
It's just kind of anticlimactic.
What?
It's just kind of anticlimactic.
Yeah. Well, the Borneo story, that's a 20-year story. I mean, you can't do that now. So I acknowledge my postdocs, particularly when I first came to LSU. Mark Hafner had raised this huge grant, which helped pay for Peter Dunn and Linda Whittingham, who got me started here at LSU. Hugely helpful. My grad students have been superstars. Of course, they've done all the work. That's why I don't know the top from the bottom of a pipette. And of course, Jody, you put up with my tamper fights with Sibley and with Vincent Sarich and quitting jobs and graduate school and all that kind of thing. So that's it. Thank you very much. I thought we'd go drink beer.
We're going to take some questions. We'll take a few questions, but then you can ask Fred during the beer soda social.
Is there a way to turn on the lights here? Yeah, oh, that's nice. Yeah.
So I'm curious about what the distance people, what the Sydneys were saying about Willie Hennig. He had brought up the ideas of synapomorphies, but 30, 40 years before they were using these distances commonly, and were they just ignoring the idea of synapomorphies until likelihood came along? Until Felsenstein came on the picture and started criticizing them.
No, just that Sibley never paid it. Sibley and Wilson and those guys just didn't think that they knew that morphology was just way too plastic to be of use. That's how they felt. And so they just produced distances to study relationships. They ignored the Hennigians. But then in the 80s, the Hennigians started complaining a lot about these things. I mean, they were on the verge of extinction, and they were fighting like tigers to save themselves. And so that's when this story about distances and apomorphy came up.
Sorry, Dennis, could you repeat the question so that people on Zoom can hear?
Oh, well,
And I
the
just had a
question
request.
was, I mean, basically it was, did the distance people just ignore the Hennigian point of view? And the answer is yes. I mean, the thing is that Hennigians, cladists, thought that they called themselves phylogenetic systematists. They didn't think that the molecular stuff was phylogenetic systematics, whereas the molecular people thought they were doing phylogenetics. They were. They just had, so that the two minds didn't meet.
You got a question for Mary Jane on the thing.
I do.
Yeah, first I'd like to say hi to Frank Gill, who's on this Zoom, and we were buddies at Michigan in grad school. So hi, Frank. Fred, I'm sure you won't mind if I point out certain absurdities in your talk.
Absurdities?
I mean, absurdities. Yeah, leaving aside the thing about the bats. So at the beginning, you had three reasons why you thought molecules were wonderful. And one of them was it was close to the genes. And so, because phenotypes are difficult to interpret, and then you kept going. First of all, you talked a whole bunch about molecules that are phenotypes, like proteins and other things. But then you got really excited because you got to using DNA, and you were really on the genes. And I thought, as I listened to you talk, that the further you went along, the further you got from biology. One of the things about this is you mainly just got into phylogenies, and you got into problems like how to explain rates of evolution. You have to do phenotypes to figure that out. And then
No, you don't.
And then finally, well, if you want to, let me, okay, here's what I mean. All right, here's what I mean. I'll say it in terms of phylogenies. If you look at a phylogeny, every branch is a speciation event. And in order to understand the biology of the dynamics under that phylogeny, you have to understand speciation. In order to understand speciation, you have to understand phenotypes. you can have one genetic change or even none, and you can have a phenotypic, very important determinant of speciation phenotype. Because if it affects inability to mate, you can have speciation with very little genetic change, very little genetic distance.
Okay,
Anyway.
I understand. I understand. So you're defending the phenotypic view, and I'm not arguing against the phenotypic view. I mean, if you're trying to, we're building phylogenies, and if we're building phylogenies, using phenotype doesn't work. So you've got to do what you've got to do. What you've got to do.
Yeah. Well, at the end, you sort of vindicated your existence by bringing in biological questions that you could use phylogenies to help understand.
Well, yeah. So we recognize the point of the process, but the big battle at the beginning was to get the tree that explained genealogical relationships. And the only way we've been able to do that, and it's now been verified over the last 20 years, is by comparing molecules, not by comparing phenotypes.
Yeah, I see what you've done, but I also see, look at what goes on in museums. I mean, luckily LSU has a really good active museum. As you and most everybody here realizes, museums have gone down the drain. And especially alpha taxonomy has gone down the drain. So that means that understanding species, I mean, you kind of waved off.
Mary, the people here can't hear you. So I'm going to have to, we'll have to have this conversation at another point.
Yeah, I didn't realize, but you could hear me and others.
I can hear you.
You mean people there in the auditorium. Okay, well, we should do this anyway, aside.
Okay, thanks.
Let's do one more question and then from the audience, and then we'll break.
One more question from the audience.
So Fred, looking back on your many decades of experience, it's one of the cool things about this. It strikes me that you got to see the evolution of an entire field and be a part of it. So I'm wondering if you could predict the future of the next 10 or 20 years. What are you most excited about?
Well, the question was a statement that I had seen, the field grow to the current point, and basically where is it going to go? The interesting thing is that when I was coming up, I knew everybody in the field, almost everybody. It was a very small universe. And now it's gigantic. I mean, exploded with all that comparative biology in the 90s. And now with genomics, it's just going crazy. So I personally think the simple problems of systematics are almost all solved for birds, and will soon be solved for most other vertebrate groups. And the real future is population genetics to understand what's going on at a closer level, and then of course everything else. So it's just exploding. What's that?
Everything else.
Everything else is all the other studies of the world, ecology studies, behavior studies.
Before everyone breaks, I want to present to Fred a small gift that many people contributed to. Friends, colleagues, students, perhaps some enemies or happen to be retiring. I'm not really sure. They're all listed in this card. The wrapping job is mine. I take full credit for it, and I'll pull it out of this box for you, Fred, just because I was the one that wrapped it. It's not very well wrapped.
I haven't left yet. I'm still.
I'm so sorry. The people on Zoom asked to be able to see, but I don't know how to quite.
Whoa, look at that. I don't know if you guys.
Dan, are you here? You were here. There's Dan. Stand up, Dan. Dan. For this painting, and this is a temporary frame. You and Jody can figure out a better frame to hang up in your house. Right?
That is really cool. That is the The pain, remember that old commercial? Fitz, remember that commercial, the heartbreak of psoriasis or pityriasis? The heartbreak of pityriasis.
Yeah, it's the
That's
heartbreak
Bornean-
of pityriasis now, right?
Yeah, that's right. That's the Bornean bristlehead. That's great. Thank you very much, Dan and everybody. That's
All right, we're breaking. We're gonna stay in here. Is that correct? We're staying in.
Can
here.
I, Fred, can I jump in?
Stick around. Some of you I know need to leave. Those of you who want to stick around for a beer or a soda. We'll see the rest of you over at the annex for the jambalaya dinner for the. Thank you all for coming. Really
Hey,
appreciate it.
Fred.
Thank you, Fred.
Can you hear me, Fred? Hey, Fred? Can you hear me?
I just wanted to make sure you didn't close out of your Zoom because there are a few.
I'm not
people
gonna,
who
I'm not
want-
gonna, I'm not gonna close out of the Zoom,
Okay,
but I
I'm
wanna
s-
save,
Okay.
I'm gonna save the, where's the slide? Stop recording.